Thank you, Neil, and my apology. Neil is, of course, Goodman's dad, not Goodman's son, as I initially described him. Our second speaker is Kevin Clayton-Greene, who is going to give us a view on this pest from an Australian perspective. And if we can store up all the questions for the end, we should have enough time for a decent question-and-answer session then. Thank you.
Thank you very much, Robert. And first of all, thank you very much to the World Potato Congress for inviting us here to talk to you today about zebra chip and biosecurity. What I'd like to do now, is to, first of all, just give a bit of an overview as to how we perceive zebra chip from Australia, or the complex, and a bit of an overview from a biosecurity perspective. Talk about a couple of the issues that really make it I guess a good case study as to biosecurity, and then raise some of the issues that I think zebra chip and generally raises about biosecurity as a whole, not just about this complex, because it's a very complex area. So what do I mean by plant health today? Well, in the context of today's talk, I'm going to strictly speak to you about it from a biosecurity aspect. In other words, pre-border entry into a area or region or country, and I'm not going to consider what other contexts that people would normally talk about with plant health. What I'd like to do is that we don't, in Australia, we're lucky, we don't have zebra chip. Our cousins across the Tasman Sea do, and it's in the response that happened in New Zealand, and I guess where it's happened elsewhere in the world, and Neil's touched on that. The initial impact of this complex in any region has usually been devastating. And one also should remember that it's not just potatoes this complex affects, it affects tomatoes and most solanaceous crops to a greater or lesser extent. So it has a devastating effect when it first appears in any given area. So what did we do in Australia when we found out about it? It first became discovered in New Zealand in 2006. I think it was found in a nursery or greenhouse. And about 2 years later, we really became aware that it was a major problem. So why should we be concerned about it in Australia? We're an island. We're a long way from anywhere. The nearest TPP is in New Zealand, which is still 2,500 kilometres away. The winds blow from west to east, so it shouldn't get to us. We all think that the government looks after our quarantine, and it's their responsibility. I want to address some of these issues first off as a way of setting the scene. This puts Australia in perspective, where we are in the world, and it's probably of interest to note for our European cousins that it covers most of Europe, and in fact the distance I think from Perth to Melbourne is similar from Paris to Moscow. So it's a big country, but it's a long way away from anywhere. However, in 2006 lettuce aphid arrived in Australia from New Zealand, and they don't fly. And within 3 months they were up the entire eastern seaboard of Australia from Melbourne up to Brisbane. It's a government responsibility. Well, the problem in Australia is that 90% of Australians live in capital cities or large cities. The rest of us who live in rural electorates, we only make up a very tiny percentage of any government, so they're not really interested in what happens in rural electorates. As a result, biosecurity in Australia, as it probably is in many countries, is a soft target for budget cuts. It's boring, nobody finds it very interesting, and it would be fair to say that most of the population is completely disengaged from it. And to give you an example of why it's such an issue, in the last 15 years, through the Port of Melbourne, which is Australia's biggest seaport, sea cargo movements have increased by about 250%. Biosecurity funding, or anything related to that, has actually decreased in real terms, let alone kept up with it. And horticulture and agriculture, in terms of Australia, is a very, very low priority. Only about 3% of Australia's population is involved in agriculture or horticulture. Well, people think we've got a good quarantine system in Australia. You get your baggage x-rayed, there's dogs at various ports and so forth. Well, New Zealand's also got a really good quarantine system, and the psyllid got there, and it crossed the Pacific, which is about 10,000 kilometres of ocean. We only need to look at the spread of citrus psyllid in the US to see how quickly these pests can spread. And a major issue that's just virtually wiped out the entire Florida citrus industry, and now California is fighting an action now to try to prevent it doing the same there. PSA in New Zealand has devastated the kiwi fruit industry. And we're looking now at the issues with Dickeya solani in Europe. It's also worth remembering that biosecurity, insofar as it's interpreted by most governments who've signed up to the World Trade Organisation, it's risk-based. There is absolutely no such thing as zero risk. And in Australia alone, we have several thousand interceptions a year at the border, and we have about 30 or 40 incursions of exotic pests. The other argument that the wind's blowing the other direction, well, they don't always, and this is a graph showing the meteorological conditions in August last year, with a large low blocking in the Tasman Sea, and you can see that the winds are actually coming to Australia from New Zealand in the upper atmosphere. And that's believed to be how lettuce aphid got to Australia as well. So in preparing for incursions, there's a number of issues that we have to look at. The first is that most incursions are generally reported by a grower or the general public. Now, the case of zebra chip, it's a very beguiling disorder. You've seen from Neil's slides, yellow leaves, tuber discolorations, all of those things you can see in any potato paddock without necessarily having zebra chip. So it's very similar to lots of other things. So if you don't have your eyes and ears about you, you could easily finish up with having an infestation without even knowing it. The other key thing in managing any biosecurity is that one has a rapid response and identification system, which Australia didn't have. And then there's three other issues there which I've highlighted in italics because they're very important, but I don't think we've got a good handle on them, and I don't think most people do, which is the social consequences for a person making a report, the public relations side of it, how you manage it, given that most people live in cities and don't want any insecticides around them, and how you then go on to manage the incursion from there on. So what did we do in Australia? Well, really, industry has to take the lead. It's not going to come from government. They don't want to know, and they don't care, unless they really have to, and it's a vitally important electorate. Otherwise, I have to say that governments really aren't that interested in managing biosecurity incursions. So in 2009, the National Vegetable Industry Body, AusVeg, organised a summit whereby we assembled people from both New Zealand but also a range of people in Australia to look at what we could do to prepare ourselves, because the general consensus was that at some point in time this disease is going to finish up in Australia, so we may as well get ready for it. One of the outcomes of that was that we asked the New Zealand people if we could have observer status and be involved at seeing how they were managing the disease through their scientific committee. And they very graciously allowed us to do that, which greatly helped us get some preparedness on the ground in Australia. Out of that, we developed and used some of the diagnostic protocols so that at least we had the primers and the technology in Australia for a rapid identification, which meant that if something does happen, we don't have to send it overseas and have the whole place in shutdown mode for 2 weeks while we wait for an identification. There was also a monitoring program set up along the eastern seaboard of Australia, which is ongoing. And we produced a range of publicity materials, including laminated ute guides, we call them, things that you put in your truck or whatever it is that you're on the farm, so that people have got a ready-made handle for looking at the sort of typical symptoms. And there was regular updates and seminars produced. However, despite all of that, and most people are aware of it, the general level of awareness of what to look for is pretty poor. And one of the great issues that comes out of this is that how do you maintain awareness about something that might be here tomorrow, or it might be in 10 years' time, and you've got no way of predicting it. And that's a really fundamental issue, because most people, most growers, we're too busy concentrating on our day-to-day things, and we really don't want extra problems. So if we see a problem in the paddock, it's human nature to suggest, well, it's probably something that's just there, and we don't really want to make any life difficult for ourselves by going and finding out that it's something bad. So I think the lesson that we learn out of this is that there are a number of drivers for biosecurity. The first one is trade. The second one is trade. And the third one is trade. And that's really what drives our biosecurity system in Australia. And I have to say, I think from what I've talked to other people, it's pretty much the same around the world. And that has some really severe repercussions for people involved in the sphere. The first one is that we need to engage with the wider public and government. I've already touched on the fact that most people really don't care, they're not interested, they don't even understand what biosecurity is about, or quarantine. The level of awareness and knowledge about that is very, very poor. I mean, I find it amazing. I came to the UK, my baggage got checked through to Edinburgh. Nobody asked me about it. There could have been a bag full of potatoes in there, full of all sorts of wonderful diseases, which could be now. Hello, what's happened here? The second issue that comes out is that those who benefit from trade, which is what most signatories to the World Trade Organisation do, are not necessarily those for whom it poses a risk. So the people who actually want to do the trading aren't necessarily those that are going to be impacted by any of the negative effects that come out of any incursion. The social cost, and this is something that's more or less completely ignored. And by that I mean that when somebody puts their hand up and says, I've got a problem, they can really face social ostracisation in their community. We've seen that in Australia with outbreaks of PCN, where people put their hand up, they get death threats. Their kids get hounded out of school. And governments don't appreciate that. They think that people will do that out of the goodness of their hearts. Well, if you're facing the risk of being sent out of your community, where you've lived and grown all your life, by putting your hand up and indicating you've got a problem, a lot of people, and most people, would think twice. And we really haven't addressed that in the area of biosecurity. The other one about who pays, the private versus public good argument. This is a classic. The conventional view amongst regulators is that the more an industry is worth to the private people involved in it, the more they should pay. So in other words, if it's worth a lot of money to a whole lot of people, they should be paying for it, not the government. And that comes out of better cost-benefit analysis models. And for every problem you can throw up, you can develop a cost-benefit analysis, and it'll show you pretty much what you want it to show at the end of the day. The other big issue is disentangling the biosecurity argument from trade protectionism. And unfortunately, all too often, we see biosecurity being used as a protectionist method, which it should not be. But we shouldn't allow the fact that it's a bit like racism. I can say something as a white person, and I can say something as a black person. But if I say it about the other race, it's regarded as racism, whether it's true or not. And the same goes for biosecurity. We can talk about it ourselves, and we can talk about it, but as soon as we start talking about protecting your borders from incursions, the bioprotectionist argument gets raised. And we haven't really addressed that, because it's politically expedient for growers, for whoever is impacted by this, to use it and trot it out when they want to. And that has to be disentangled from the biosecurity argument. The other one that we face is, I've called it regulatory hubris. The people that make the rules and decide things have got a very, very poor understanding of industry. They don't understand the operating imperatives of industry, and what's more, they show a great reluctance to listen to them. A new one now that I think is also an important issue that comes into this, and it relates to sequencing and all the PCR techniques now. We've got to the point now where we can find virtually anything. What we don't know, and what we're not smart enough to know, is when is a new sequence a new pest or a disease, and what's its likely impact to be. And the other side of that argument is that many scientists like to go out and publish papers, and you can generate a lot of papers with PCR sequencing very quickly. Suddenly finding a whole suite of viruses or whatever in various organisms that you test around the country, which then, if they're published, become a record for that country and then create an issue in terms of negotiating trade agreements and biosecurity. And this is a very real issue that we're facing in Australia. How do you deal with that? You can't stop people from publishing. On the other hand, you don't want them to be going around shooting you in the foot. And lastly, these issues themselves, I believe, are a long-term project, and I've put them up there because I hope that I'd like some ideas about how you handle them, because there's a lot of things to be learned. Thank you very much.
Thank you, Kevin. That was a very insightful examination of the quandary of the war between biosecurity and free trade. We have 5 or 6 minutes now. I can open the floor for some questions. If you'd just like to hold your hand up, we can get your microphone over. Does anybody want to shoot first? Nobody? Yes, please. This lady.
Hairy nightshade is a weed that is becoming very bad in Ontario, Canada, and it's supposed to be a good host, right, of this Liberibacter?
Yeah, the question was about whether or not nightshade, and there we've reported that all of the nightshades, basically, that we have in the Texas area are susceptible hosts. However, what's really unique is that they don't necessarily harbor the bacterium for a long period of time. In other words, particularly with the black nightshade or the hairy nightshade, cutleaf nightshade, those are annuals. That infection dies off when the plant dies. We fully expected silverleaf nightshade in Texas to be, which is a perennial host, to harbor the bacterium for a long period of time. But we go back and sample in the same georeferenced areas. From one year to the next, the bacterium can disappear. So they are a host. We don't know if they're a significant reservoir of inoculum that's significant for the epidemiology of the disease at this point in time. That's probably different, though, after talking to Andrew Pittman about what's going on in New Zealand. So I think it is a potential. We just don't know how great it is yet.
Anyone else? Yes, please.
This is really just for information. There is an EU project. It's starting. It's just started, and that will actually look at the, in terms of the haplotype C of LSO that infects carrot. Will it also infect potato? And it's also going to look at the psyllid as well to see whether the carrot psyllid. In fact, potatoes are also a host of the carrot psyllid. So just for information. That's good to know, Colin. I think it's key to know that, to point out that I don't think that those two psyllid species that solanaceous host, potato, have to be an actual host where they would complete their life cycle, because what's important in introducing the pathogen into fields are the adults, and they all have to probe for a short period of time, and they can transmit the pathogen. So, yeah, of course, you're quite right.
Please, Gordon, this gentleman here.
Yes, you said you have some varieties, susceptibilities. Can you tell us if some varieties are immune or resistant, and if the resistant variety can carry on the bacteria?
That's a good question. We have a concerted, when ZiberChip became really important, myself and two of my colleagues wrote a grant to the federal government, and we got $10 million for a 5-year period to do a concerted amount of research on this. Part of that is to look at germplasm, screen species, and whatnot. I can tell you that we do have in Solanum tuberosum, we have tolerance. We don't have resistance. They harbor the bacterium at relatively high populations as a susceptible variety would, but there is a general absence of symptom development, like the tubers I showed with the flecking and the medullary streaking, and you don't get the finished product having the caramelization, you know, for the defect. So we don't have resistance yet, but we don't have no immunity. We do have some resistance in some species, but in Solanum tuberosum, we've only found tolerance.
Thank you, Sue.
We want to know if do you have some information about the control of the bacteria inside of the plant? Because it's more easy control, I believe it's more easy control the bacteria like the psyllid in the field.
I'm not sure I understood the question.
The question is there is it's a chance to have a control of the disease, controlling the bacteria that is carried by the psyllid.
I think
once it's control in the plant, is there a way to?
Yes.
At the current time, no. However, some great work has been done with the HLB citrus screening, which is an analogous. It's Libiribacter asiaticus in citrus, and they have a new generation of antibacterial peptides that are proving to be very efficacious in eradicating that bacterium in citrus. The difference, of course, is that bacterium increases very, very slowly over time in that woody plant, as opposed to very rapidly in potato plants. But so the answer is currently not yet, but there's some hope that there might be some products in the near future. Good question.
One more, I think.
You mentioned when you had slides up about the effect of the seed, you saw a small plant affected in the seed, and you said it's not too much of an issue at the moment. Can you just elaborate on that, please?
Well, the New Zealanders have done some work, and Andrew is here. I think their conclusions is that seed could still be an issue, but in our case, we did our work through basically quantitative epidemiology, and that is tracking in fields that had seed-borne inoculum, tracking every single one of those infections, and then tracking this is over a 2-year period, the development of the zebra chip epidemic in the field, and it was not at all related to the seed-borne inoculum. It was related to the influx of psyllids carrying the bacterium into the field. So, and the other, we very rarely, even though those plants come from zebra chip infected tubers, the titer of the bacterium is extremely low and usually undetectable. And some work that hasn't been published yet has shown that when psyllids were given the choice of a zebra chip infected plant that came from seed versus a healthy plant, that 90+% of the time they go to the healthy plant. They're not attracted to the one that has a seed-borne infection. That's not published yet, but I think that all of those pieces of information suggest that seed-borne inoculum is not epidemiologically important. And only about, depending on the variety, 20 to 40% of the tubers that have zebra chip in the tuber actually germinate anyway. Most of them, the eyes are dead and they don't germinate.
Just when I thought the seed growers could relax as well. Right, okay, I think we have to wind it up there, everybody. I'd like to thank Kevin and Neil for their excellent presentations, and thank you, the audience, for your participation. Thank you very much.